Good afternoon, everyone. I hope you're having a good Saturday afternoon. Welcome to the application section of this workshop, which will be presented. At least the first application here is based on image-based plant phenotyping and why we need distributed deep learning to accomplish some of the large data and large model challenges that we're facing. I am Bhaskar Ganapati Subramanian. I'm a faculty at Iowa State University, and with me is Kaushik Nagasubramanian, who is a senior graduate student in my group. This work is done in collaboration with several groups, including the Pat Schnabel group at Iowa State, Dan Nettleton and Yumo Chi's group at Iowa State, as well as James Schnabel's group at University of Nebraska-Lincoln. And this work would not have been possible without the help and the toolkit that RocketML has provided us. So before we get in the thick of this application, I want to take a minute or so to give a quick overview of what my group does here. So I run a computational sustainability group at Iowa State University, where essentially we are a computational group with broad interest in sustainability applications. So we have a diverse set of application foci, including energy, health, water, and food. And what we essentially do is we develop and deploy computational tools, and more recently, machine learning and deep learning tools for assessment, for understanding, for control, and for design of complex systems. So here, we need tools that can scale up. We need tools that can look at a lot of information in a rapid way, and then we need these tools to be high fidelity. Some examples are illustrated at the bottom. The left picture shows some of our recent work on doing very rapid risk assessment by evaluating the thermal and fluid flow in classrooms to look at pandemic COVID risk. The figure in the middle shows some of our recent work on looking at developing computational methods that can help assess and help us design new materials that can be used for better water desalination. And the figure on the right is the focus of this application presentation, where we look at machine learning and deep learning tools applied to food and agriculture. Machine learning, in particular deep learning, has become a game changer in agriculture. Even as recently as 10 years ago, it was almost impossible for an agronomist or a plant scientist to look at anything other than the final yield of a plant, especially when you're looking at very large areas as seen in the picture on the left. So all you could get in a reasonable amount of time using reasonable amount of effort was looking at the final product that comes out of the plant. However, just like humans, right? So plants also have a development cycle, and depending on cues and stimuli and management and love and care, if you will, at different parts of the production of the growth cycle, they provide varied and very varied amounts of yield. So the ability to actually figure out what is happening at different parts of the growth cycle can transform agriculture, and until now it has been impossible to do without the use of machine learning. So machine learning has been a step change, a transformer in terms of the ability for us to extract traits, and that's what we call phenotyping, and that's the title of the application, especially in large field experiments. The experiment here that you see on the left is done by our collaborator, Pat Schnabel's group, where they have a very large area covered by different genotypes or different types of plants, and they are repeating this experiment across multiple geographic regions, across multiple years. And what you see here in our solar cells that essentially are used to charge cameras, since this is in a place where there's not good electric power or internet capability, and these cameras take images every 10-20 minutes across the whole growth cycle. So just imagine the ability to see essentially how a plant is growing, and then you can extract out critical developmental traits, right? So, and this is a game changer in terms of linking the physiology, right? How does a plant grow? How does its genetics impact how it grows? How does crop management affect how it grows? How does environment, because it is repeated multiple years, how does the weather affect how it grows, and so on, and help us tease out features that breeders and growers can essentially optimize and perform precision control over to get us the next generation of crops that are resilient to climate change. So here is again the specifics of the experiment from which we want to apply distributed deep learning to extract out features. This is, like I had alluded to before, a time-lapse photography over the complete growing season. So about 8 to 9 weeks of growth. You have a camera that's facing one row of plants as seen in the previous slide, and this essentially takes images every 10 minutes. And this experiment is repeated across multiple locations in the Midwest, as well as across multiple years in the Midwest. And so essentially, we have about greater than 4 million images, and these are high-resolution images, about 4K by 4K images, from around 2,200 cameras. This represents a diversity of genotype, diversity of weather conditions, diversity of crop management, and diversity of soil types. And so this is an extremely rich data that is waiting for analysis. But the key bottleneck, and what kind of analysis are our plant science collaborators most interested in? The two most impactful ones are essentially understanding how the plant grows as a function of time. That's a figure on the top right there. We want to identify plant height as a function of time, and this allows us to figure out how much of biomass is the plant producing, how it's partitioning, how efficient is it growing, how efficient is it using nutrients, how efficiently is it using water. Another thing that we are also interested in is shown in the bottom right figure, where we are interested in figuring out what is the leaf appearance rate, right? So how many leaves appear as a function of time? Why is this important? This is directly related to how efficiently is the plant able to convert photosynthesis, right? So utilize photosynthesis. The more the surface area of the leaves, the more photosynthesis, and also how much it can partition into growth stages as well as into product. That is the final. Now, if we want to evaluate, here is a picture here's a video of how of one single camera that's looking at one row of plants across the growing season. So if you want to extract these traits that I alluded to in the last slide, that is basically height as a function of time for each and every plant that you see here, as well as the number of leaves as a function of time. The first step here is what this whole workshop is about, the idea of, can we do semantic segmentation, right? So that is, without going too deep into this and reiterating what has already been talked about, you're given an input image, and you go pixel by pixel, and you label what class each image belongs to, what class each pixel belongs to. So in the context of the plant science application, the image phenotyping application that we are working on, this corresponds to going pixel by pixel in the image and marking each pixel as either belonging to the background or belonging to a plant. Now you can actually see clearly in the figure on the right, it's going to be incredibly painful, incredibly difficult to actually annotate a large enough data set that can be used for training. And this is our basic challenge, right? So the segmentation, to perform good segmentation using large models, you need a lot of data, right? Using at least the current conventional methods. And to do this, to get accurate pixel level annotation, this is very laborious process. We have tried it and we have several papers that show that even when we try it out using crowdsourcing, there's a lot of inter and inter-rater variability that makes it very error prone. This is also expensive if you want to do it well. And even if you're willing to pay, this is also time consuming to do. And so we build upon recent work where we rely on artificially creating annotated data, right? So this is phenotyping in the wild, right? So because in the video that presumably you noticed, you see a lot of dew, you see a lot of wind, you see different illumination conditions, you see a lot of rain as through the season you're taking images. And so what creating robust segmentation models requires creating good annotation data. And the way we create this annotation data is actually utilize plants grown indoor and or plants grown synthetically or created synthetically indoors, and then we transfer that onto the field and create a highly realistic synthetic data set that can be used for training these models. I hand it over at this point to Kaushik, who will dive deep into the methods.
Thanks, Bhaskar. To overcome the data annotation challenges for training semantic segmentation models, we will be using two different training data creation methods. For the first method, we will use greenhouse images for the training data creation. For the second method, we will use 3D virtual plant models for the data creation. We used the RocketML distributed deep learning framework to train our semantic segmentation models. We trained UNet and PSPNet models for semantic segmentation with two different backbones for each of these synthetically created datasets. ResNet 18 and ResNet 152 are the two different backbones that we used. The first question we studied was, how can greenhouse images be used as training dataset for segmentation of maize plants in the field? This greenhouse training dataset was collected by James Schnabel Group at University of Nebraska-Lincoln. The training dataset consists of time-lapse images of greenhouse-grown maize plants belonging to multiple genotypes as shown in the video. 122,290 images were collected in total. The infield test dataset was collected at Iowa State by Professor Pat Schnabel Group. To segment the greenhouse maize plants, we first identified the location of maize plant automatically using RGB-based thresholding, and then used HSV color-based thresholding for the segmentation of the maize plants. In this video, the left side of the video shows the automatically extracted region of interest, and the right side of the video shows the segmented plant. Since the segmentation was done using indoor images, it was pretty accurate. The video here shows skeleton annotation of infield maize images. We used this skeleton annotation for our data augmentation pipeline. 250 skeleton images from 4 different cameras were used for our data augmentation pipeline. A red line was drawn over a plant stalk and a blue line was drawn over the leaves to extract the pixel information of the stalk and the leaves. We applied color and shade transfer based augmentation method for our data creation pipeline. The goal of color and shade transfer is to convert the color and shade components of the input images similar to the color and shade component of the style images. Using the color and shade of the skeleton annotated style images, we were able to make the color and shade component of the indoor images look very similar to the infield images. This process helps us in reducing the distribution shift between the indoor and outdoor plants. We used cut and paste based data augmentation method to create synthetic training dataset. In the video on the left side, we are seeing images of an empty field. These images were selected for the diversity in illumination conditions. After augmenting the indoor plants using color transfer data based data augmentation, we randomly inserted them into an empty field as shown in the right video. The plants were randomly scaled into 10 different sizes before being inserted into the field. In total, we created 85,824 images for training data and 21,459 images for the validation data. We used UNet architecture for semantic segmentation. It consists of an encoding path and a decoding path. The encoding path follows a typical architecture of a convolutional neural network. A series of convolution and pooling layers downsamples the image resolution, and the decoding path upsamples the features back to the original resolution. The input to the model was synthetically created infield maize images. The output score was 1 for the maize class and 0 for the background class. We used binary cross entropy as the loss function. PSPNet, which stands for Pyramid Scene Parsing Network, was the second model that we tried. It is a semantic segmentation model that utilizes a pyramid parsing module to extract global information. So the local and global features extracted from the pyramid pooling module helps in getting an accurate segmentation. The input was same synthetic infield Maize images, and the output was Maize or background class. The loss was binary cross entropy. For more details on this architecture, please refer to the paper. Now that we have created our synthetic data, we used CPUs for our semantic segmentation models. So why do we use CPUs for training our semantic segmentation models? Because to overcome the memory bottleneck of the GPUs, training with CPUs enables us with using high image resolution, large batch size, large models, and large amount of data in a reasonable amount of time. Training with high image resolution was important for extracting phenotypic traits like leaf angle and leaf count. Training large models on large amounts of data helps in obtaining accurate semantic segmentation models. This motivated us to use distributed deep learning approach for our problem. For the training data created using greenhouse images, we used UNet and PSPNet models for semantic segmentation. Here we show the UNet and PSPNet model hyperparameters that were used. We used RocketML distributed deep learning framework for our model implementation in the Bridges 2 PSC cluster. The input resolution for UNet was 1024 by 1024, and the resolution was 1056 by 1056 for the PSPNet. The UNet and PSPNet models were trained with two different backbones, ResNet 18 and ResNet 152. The two models with ResNet 18 backbone were trained on 2 nodes and 8 processes. There were 4 processes in each node. All the 4 different models were trained for 5364 iterations. We used a batch size of 64 and Adam optimizer with a learning rate of 0.0003. Here we show the peak CPU memory used for the 2 different segmentation models. For the UNet model, the ResNet 18 backbone used 137 GB memory, and the ResNet 152 backbone used 250 GB of memory. For the PSPNet model, the ResNet 18 backbone used 96 GB of memory, and the ResNet 152 backbone used 225.6 GB of memory. All the 4 different segmentation models used significantly more memory than the 32 GB memory available on a single V100 GPU as shown in the figure. In this slide, we are seeing the segmentation predictions of a UNet model on time-lapse images of a single camera. On the left, we show the segmentation output for the UNet model with ResNet 18 backbone, and on the right, the predictions are shown for the ResNet 152 backbone. Both models provided good segmentation predictions. The mean IOU was around 83% for both the models when evaluated on 100 images. Here we are seeing the segmentation predictions of a PSPNet model. Again, both the models provided good segmentation predictions. The mean IOU was around 84% for the ResNet 152 backbone and 82% for the ResNet 18 backbone. The larger ResNet 152 backbone was helpful in getting better accuracy. Since we were able to get good segmentation output using greenhouse images based data augmentation, we asked if we could use virtual 3D plant models for segmenting infield images. Our collaborator James Nabel at University of Nebraska-Lincoln was able to create these 3D maize plant models with different shapes using Plant Factory software. These 3D plant models were then projected into 2D images using Blender for creating synthetic training data. We can use this software to create plants with different phenotypic traits like plant height and leaf numbers, leaf angle, etc. Similar to our greenhouse-based data creation pipeline, here also we applied the color and shade transfer method for the virtual plant. The video shows a single virtual plant translated into different color and shade components based on the skeleton type. So this helps us in reducing the distribution shift between the indoor and outdoor images for the virtual plants. The video here shows the virtual plants after using the cut and paste method based data augmentation. We inserted the virtual plants randomly into empty background to simulate the infield data. In total, we created 6,496 images for training and 1,624 images for validation. For the training data created using 3D virtual plant, we again used the UNet and PSPNet models for semantic segmentation. Here we show the UNet and PSPNet model hyperparameters that were used for the segmentation. The input resolution was same as before, 1024 by 1024 for UNet model and 1056 by 1056 for the PSPNet model. The UNet and PSPNet models were trained with two different backbones as earlier, with ResNet 18 and ResNet 152 networks. The two models with ResNet 18 backbone were trained on 2 nodes and 8 processors. The UNet model with ResNet 18 backbone was trained for 6,000 iterations. The UNet model with ResNet 152 backbone was trained for 3,000 iterations. The PSPNet model with ResNet 18 backbone was trained for 10,000 iterations. And the model with the ResNet 152 backbone was trained for 5,000 iterations. We used a batch size of 64 and used Adam optimizer with a learning rate of 0.0003. The memory usage was similar to the models used for greenhouse images. All the 4 models used significantly more memory than 32 GB memory, which is commonly available on single GPU. On the left, we show the segmentation output for the UNet model with ResNet 18 backbone. And on the right, the prediction is shown for the ResNet 152 backbone. Both the models provided good segmentation predictions. The mean IOU was around 82% for the ResNet 152 model and 78% for the ResNet 18 model. The larger backbone was helpful in getting better performance. Although PSP ResNet 152 model performed better than UNet model for the data created using greenhouse images, they performed poorly when compared to UNet when trained using synthetic images as seen in this video. The ResNet 18 model performance was 68.9% and the ResNet 152 model performance was 78.79%. In conclusion, semantic segmentation of complex field data needs large amount of annotated data. The greenhouse and virtual Maize plants were utilized to create large amount of training data. UNet with ResNet 152 backbone was the best model for greenhouse images. The PSPNet with ResNet 152 backbone was the best model for synthetic images. Distributed deep learning was critical to train large semantic segmentation models with high image resolution and large data in reasonable amount of time. In future, we plan to use the synthetic data creation pipeline for instance segmentation and extend the distributed deep learning framework for self-supervised pre-training with large amount of unlabeled data. Thank you for attending our talk. We can take some questions now.